अस्सलामु अलैकुम। आऊज़ु बिल्लाहि मिनश्शैतानिर रजीम। बिस्मिल्लाहिर रहमानिर रहीम। कुल हुवल्लाहु अहद अल्लाहुस समद लम यलिद व लम यूलद व लम यकुल लहू कुफुवन अहद। कल हमने थोड़ा सा इंट्रोडक्शन टू फार्मास्यूटिकल एनालिसिस पढ़ा था और कोशिश की थी कि हम फार्मास्यूटिकल एनालिसिस को डिफ़ाइन कर सकें या उसकी इम्पोर्टेंस को जान सकें। मैं थोड़ा सा फिर रिवाइज कर देता हूँ कि फार्मास्यूटिकल एनालिसिस इज़ ब्रॉडर टर्म विच कैन डिफाइंड इन मैनी वेज. It is the series of process that are used for identification, determination, separation, purification, and structure elucidation of given compound used in the formulation of pharmaceutical product. The components to which the pharmaceutical analysis is done are normally the active pharmaceutical ingredient, pharmaceutical excipients, and the contaminations present in the pharmaceutical products are the drug metabolites. The pharmaceutical analysis, the samples are typically finished pharmaceutical drug products, and you know very well that what is a drug product. Drug product is a dosage form ready to use. For example, tablet is a drug product, injectables are drug product, capsules are a drug product, a dosage form that is ready to use. You know that there are different type of dosage form: oral dosage form, parenteral dosage form, topical dosage form, ophthalmic, nasals, and intrathecals. There are so many dosage form, and the drug product is the broader term that as dosage form ready to use that contains not only the therapeutic moiety but also contain the excipients and the other material required for the formulation of that drug product. Not only for the quality of drug product, not only the therapeutic moiety is important, but the quality and the quantity of excipients is also important. If, for example, previously we were discussing the manufacturing of Panadol tablets. If it doesn't contain the quality of disintegrator and the not exact the quantity of disintegrator, then what would happen? The tablet will not disintegrate or not dissolve, and when it is not disintegrated and dissolved, it will be not, the medicament will not be present on the absorption side, and ultimately the absorptions will be the slower. And when the absorption is lower, it means that plasma drug concentration is lower and the required therapeutic response will not be achieved. Similarly, the other things we learn, the type of pharmaceutical analysis we learn that the qualitative analysis and the quantitative analysis. Qualitative analysis, it is a type of pharmaceutical analysis in which non-quantifiable method of determination of what constituent of substance is present in a known sample or a compound. For example, identification of specific constituents, elements, or the functional group present in the sample. The most techniques used for the qualitative analysis are the UV analysis, IR, spectra, NMR, and C13 are the techniques that are used for the qualitative analysis. Similarly, in chromatographic, HPLC and GC and TLC are also used for the qualitative analysis. HPLC is a technique that is excellent for the qualitative and the quantitative analysis of drug. For example, identification of the specific constituent, elements, or functional group present in the sample. Identifications mean to confirm the identity of analyte in a given sample. Therefore, identification can also be referred through the qualitative analysis. For example, identification of mefenamic acid, paracetamol paracetamol, and you can diclofenac sodium in different formulation. Then we comes toward the quantitative analysis. Quantitative analysis is a type of pharmaceutical analysis which is intended to measure the exact concentration of substance of interest in the given sample. When we talk about the substance of interest, it may be the active therapeutic moiety and or it may be the excipients. For example, Poundstone, that is a tablet contains 500 milligram of mefenamic acid. So the quantity of mefenamic acid 500 in individual Poundstone tablet is determined by the pharmaceutical analysis after compression and before releasing into the packing. It means that only quantitative analysis we have to develop such a method which confirms the quantity of substance of interest in the particular dosage form. That may be the any volumetric analysis method as previously we done. That may be the spectroscopic method. That may be the any you can say other method, but it will tell us about the exact quantity of the mefenamic acid present in the Poundstone tablet. This quantitative analysis of any drug product ensures the direct plasma concentration of the drug. If the quantitative analysis confirms the presence of exact quantity of the drug in the dosage form, it means that would be available at the absorption site, and after absorption it would be available in the systemic circulation and the site of action, and the desired therapeutic response will be obtained. Previously we discussed that there are different type of pharmaceutical analysis method: physical analysis method, instrumental analysis method, and lastly we done the volumetric analysis method. When we talk about the volumetric analysis, it means that the quantitative analysis is being handled with the exact quantity, the measured quantity of analyte mean sample, which is analyzed with a standard solution. Here we come, the term is used standard solution. What is a standard solution? Previously you have learned in Pharmacy First Professional when you studied the chapter of solutions that standard solutions are those solutions which have known concentration. What does it mean? कि standard solution में आपको पता होगा how much quantity of the solute is present in that solution, and the concentration may be expressed in different way. First of all, the first method for expressing the concentration is the normality. We talk about the normal solution to normal, 0.1 normal, decinormal, centinormal, and what is a normal solution? Normal solution can be defined as that number of gram equivalent dissolved in 1000 ml of solution. For example, we have to prepare one normal solution of sodium hydroxide. Sodium hydroxide has its 1 gram equivalent equal to 40 gram. And when 40 gram of sodium hydroxide is dissolved in 1000 ml of solution, it is a 1 normal solution. The other example of the solution is that oxalic acid solution. Oxalic acid has 2 replaceable hydrogen and its molecular weight is 126. When oxalic acid is with the 2 water molecule of hydration, its molecular weight is 126 gram. Now, as you know that it has two replaceable hydrogen. It means that for its gram equivalent weight, we have to divide its molecular weight on 2. So 126 when divided by 63, then 63 gram of oxalic acid dissolved in 1000 ml of solution is known as the normal solution. This is another term used for the concentration of the solution. Then we can express the concentration of solution by molarity. Molarity, you know very well that number of mole dissolved in 1000 ml of the solution. Number of mole, for example, sodium hydroxide 1 mole is equal to 40 gram, and oxalic acid 1 mole is equal to 126 gram. When 126 gram are dissolved in 1000 ml of solution, it is the 1 molar solution of oxalic acid, and when 40 gram of sodium hydroxide is dissolved in 1000 ml of the solution, it is known as the 1 molar solution. Here the sodium hydroxide 1 molar and 1 normal solution both are the equal, because there is only one replaceable hydroxyl ion, and gram equivalent weight and molecular weight is the same. This is how we can express the concentration of the solution. इसके अलावा जो है आप ये देखें कि हमारे पास परसेंट सोल्यूशन होते हैं, वेट बाय वॉल्यूम 5% सोडियम हाइड्रोक्साइड सोल्यूशन. What does it mean? 5 gram of sodium hydroxide dissolved in 100 ml of solution. Similarly, the other concentrations methods are volume by volume, and then volume by weight, and then volume by volume solutions can be made. The other concentration terms are the ppm solution, part per million solution, and the part per million solution that we will study later on, that how these solutions are prepared and when they are utilized. We also discussed the simple methods for the volumetric analysis. Volumetric analysis is the major type of analysis that is used in the pharmaceutical industry to determine the different percentage purities of different, you can say, in samples, and that is economical as well as the reliable method for the estimation of therapeutic moieties in the given substances. For volumetric analysis, you know very well that one must assure that the weighing and measuring are the important step for the accuracy of the result. Weighing and measuring, for example, when our weighing balances are not calibrated properly, what does it mean? That the standard solutions are not prepared properly. Similarly, if the volumetric apparatus are not calibrated, then the concentration of the solution may vary. For the best result and for the accurate result, we must calibrate our weighing balances, our glasswares, and other apparatus used for the volumetric analysis. Volumetric analysis, as you know very well that in previous years you have standardized the sodium hydroxide solutions and many others unknown concentration of the given samples in the. It wholly solely is depending upon your accurate weighing and accurate, you can say that, measuring. When your measuring and weighing is accurate, you will get the excellent result for that. Chemical reaction that are usually occurs in the volumetric analysis, there are of four type, that are neutralization reaction that are used for the acid base and in aqueous and non-aqueous solvent. Number 2, for oxidation reduction. Number 3, precipitation reaction. And number 4, the complexation reaction. Each type of used extensively for the analysis of drug and dosage form. Official in USP and national formulae will be will be examined or will be testified. After it, we have to learn whatever the analytical technique is. Either it is volumetric analysis, either it is physical, either it is instrumental analysis. The main thing is the separation of component that is going to be analyzed from the drug product. For example, if we have to determine the concentration of mefenamic acid in Poundstone tablet, then we have to extract the mefenamic acid from that particular dosage form. If we are able to separate mefenamic acid from that dosage form, that would react with the you can say in volumetric analysis, that would be examined by the spectroscopic analysis. If we are able to separate, then we would be able to get the result of that drug product. There are different, you have learned in previous year many different techniques, separation techniques. For example, evaporation. एवेपोरेशन में क्या होता है कि सॉल्वेंट जो है वो एवेपोरेट हो जाता है और जो आपका वो है साइल्यूट है वो उसमें रह जाता है. सिंपल उसमें क्या होता है, फॉर एग्जांपल आपने शुगर का जो है कंसनट्रेटेड सोल्यूशन बनाया है, उसमें जो है ना आप उसको कैसे शुगर को सेपरेट करना है, आप उसमें से जो है ना थोड़ी देर टेम्परेचर पर रखेंगे, वाटर एवेपोरेट हो जाएगा और बाकी आपको क्रिस्टलाइन शुगर मिल जाएगा. ये तो एक बहुत अच्छा मेथड है. इसके अलावा आपने देखा होगा कि फिल्ट्रेशन में क्या है कि जो सॉल्यूशन फॉर्म में नहीं होते, इनसॉल्युबल मटेरियल होता है, दैट कैन बी सेपरेटेड बाय द फिल्ट्रेशन. फॉर एग्जांपल जो इनसॉल्युबल मटेरियल है, सैंड है वाटर में या अदर सॉलिड इम्प्योरिटीज़ हैं, तो हाउ कैन बी सेपरेटेड, आप उसको फ़िल्टर कर लें. फ़िल्टर में आपका सॉल्वेंट जो है वो प्योर हो जाएगा और जो सॉल्यूट में इम्प्योरिटीज़ हैं वो उसके ऊपर, इनसॉल्युबल इम्प्योरिटीज़ वो उसके फ़िल्टर पेपर के ऊपर रह जाएंगी. सिमिलरली आपने देखा होगा कि इसके अलावा हमारे पास और भी बहुत सारी सेपरेशन टेक्निक हैं. सेंट्रीफ्यूज है जो कि उसके डिफरेंट उसके मॉलिक्यूलर मासेस पे करती है. सीविंग है जो उसके पार्टिकल साइज़ पे करती है. और इसके अलावा मैग्नेटिज़म है जो कि हमारे मैग्नेट के ज़रिए हम जो है आयरन और दूसरे जो मैग्नेट से अट्रैक्ट होते हैं उन पार्टिकल को अलग कर लेते हैं. ये बहुत सारी जो है ना सेपरेशन टेक्निक्स हैं जिनके ज़रिए हम जो है अपने रिक्वायर्ड कंपोनेंट को सेपरेट कर सकते हैं. और जो सबसे मोस्ट और एफिशिएंट टेक्निक है, दैट इज़ द chromatographic technique. Chromatographic techniques basically are the techniques used for the separation of different components in the different drug product for their qualitative as well as quantitative analysis. Later on, inshallah, in that detail, we will discuss the different chromatographic techniques used for the qualitative as well as the quantitative analysis. Here, there is an assignment for you people that you have to write in your own words that how many separating techniques you know for the separation of different component. जिसमें से 3-4 मैंने जिक्र किया है. फिल्ट्रेशन है, एवेपोरेशन है, मैग्नेटिज़म है और इसके अलावा सीविंग है. इसके अलावा क्रोमेटोग्राफिक टेक्निक्स हैं और इस अपना सेंट्रीफ्यूगेशन है. तो at least you people have to write down the 10 separating techniques being used in the pharmaceutical analysis, and that you have to submit till the next class. Thank you very much. Have a blessed day, and inshallah we will remain in contact in future also. Thank you very much, and Allah Hafiz.